This is a course, a version of which I've taught almost every year for the last 20 years, and it evolves a little bit every year. I think I get a little bit better at it, so hopefully you'll get some advantage from that experience. But the idea is to try to present to you sort of what's exciting about biomedical engineering, the ways that one can take science and mathematics and apply that death from infectious diseases and parasitic diseases if you live in places other than the US or Western Europe, for example. And so the problem of infectious disease prevention and treatment isn't solved yet. You know this, right? And so there's plenty of room to still innovate in that way, to develop new methods that could protect against diseases like AIDS or diseases like malaria that we don't have problems with here, but they do in many parts of the world. And so we'll talk about that. I mentioned the book for the course. And the book is a book that I've written. It's not published yet, and so I'm going to put chapters from the book that are in fairly final form, and I think you'll find them easy to read, but you don't have to buy it. It's going to be posted on the internet, and I'll post chapters sort of in advance of the readings. And if you looked on the class's server, you saw Chapter 1, and Chapter 1 describes some of the sort of organization of biomedical engineering into subdisciplines, which I've listed here. And so we're going to talk about thinking about the body as a system that can be understood the same way a motor could be understood or a computer that could be understood. And that study is systems physiology, and that's an important subdivision of biomedical engineering. We'll talk about instrumentation a little bit, and I've mentioned this, things like the EKG machine and the heart-lung machine, or instruments that are designed to either keep patients alive or to allow you to, wasn't what I thought, or allow you to monitor their function over time. We'll talk about imaging, which I mentioned, biomechanics, or the study of humans as mechanical objects. We'll talk about a field which is growing now called biomolecular engineering, and that is the design of biomaterials, or new materials that can be implanted in the body. It's new ways of drug delivery. It's this whole field of tissue engineering that I mentioned earlier. We'll talk about artificial organs, and we'll talk about systems biology, or thinking about how to acquire information from things like gene chips and use that information to understand what's happening in a complex organism like you. Now, I've highlighted 3 of these in blue here: imaging, mechanics, and biomolecular engineering, because if you go on to study biomedical engineering here at Yale anyway, these are the things that you might pick to emphasize on. These are the things that we do best and where we have advanced coursework available in these 3 categories. And so I'm going to emphasize these 3, but we'll talk about all of these subjects as we go through the course. The syllabus is posted online. I've just copied it here so you could take a look at it. So week 1, we're trying to talk about this question: what is biomedical engineering? There's some chapters here for reading. Chapters 1, 2, and 4 I list. I've only posted chapter 1, which basically reviews the things I've talked about today. Chapters 2 and 4 are really reviews of things that you probably already know something about. So they're reviews of basic chemistry, so chemical concepts that are important for us to all understand as we move forward, and review of proteins and biochemistry, basically. So I'm going to post those online, and we're not going to talk about them directly in the lectures, but they're there as a resource. So if you read about something like pH and you've forgotten what pH is, you can go back to chapter 2, which is posted, and you can read about pH, and I try to take you through sort of what you need to know in order to understand the rest of the course material. And if you've forgotten about proteins and what their structure is like, you can go to chapter 4 and read sort of a brief review of protein biochemistry. In the section this week, I'll talk about the section meetings in just a moment, but there's no required section meeting this week. During the section times, I'll be available if you feel like you want to read chapters 2 and 4 and then come and ask questions, sort of a tutorial on these topics of chemistry and biochemistry, then I'll be available to talk about that during that time. We'll start with week 2 talking about genetic engineering. What's DNA? How can it be manipulated? How is our ability to manipulate DNA led to things like gene therapy, which can now be done in people? And so we'll talk about that, and that's what chapter 3 is about. We'll talk about cell culture engineering during week 4. How do you maintain cells in culture? What are the limits of this? How can you use cultured cells to do things? And how do engineers build new things out of cultured cells is going to be a subject we talk about throughout the rest of the course. And the chapter is listed here. So I think that's enough. You can follow along with the syllabus and see sort of what the topics are each week, what the reading assignment is to do before the lecture in order to get the most out of the lecture. Now, each week we have a section meeting, a required section. All the sections meet on Thursday afternoon. And the idea of the section is to sort of amplify on some subject we've talked about during the week. And we do this in the undergraduate biomedical engineering laboratory in the Malone building so that we can do demonstrations and sort of hands-on projects to really get a little bit deeper into the subject that we're considering. And so in the first week, we run a section called From Strawberries to Gene Therapy, where we talk about DNA, extract DNA. You can play with the DNA of an organism, and we can think about how to use DNA for other purposes. In week 3, you'll actually do some cell culture in the laboratory and look at cultured cells and learn how to do some manipulations on cells in culture, and so on throughout the weeks. We have a 1-hour section that's designed to give you some more detailed experience, some hands-on experience with some of the topics we're talking about. There are no lab reports that are due. There sometimes will be homework assignments which sort of build on what we've done during the section, but it's not a lab in that sense that it's a long experience in the afternoon or that it requires any detailed reports. But it is required and I think an important part of the course. There's a midterm exam halfway through and a final exam at the end. And there's a term paper which is due near the end of the course. So this just say a little bit more about the sections. There's 3 sections. We have online discussion sections sign up. Has anybody tried to do that yet? Just so they know that it's available. So it was supposed to be available from day 1. You can sign up for a section that fits your schedule. And this is sort of the list of things that we'll go through in the section meetings. Grading. 30% of the grade is for the midterm, 30% for the final. And so the final is not cumulative. The final covers only things for the last half of the course. So it's really just like covers half the course, but it's given during the final exam period. There's a term paper, which I'll talk more about as the weeks go on, that's also worth 30% of the grade. And you'll have approximately weekly homework assignments that account for 10% of your grade. But they have an impact beyond the 10% because if you can do the homework and you understand the homework, you're going to have no problem with the exams. So I encourage you to spend more time than their weighting would suggest. So how do you get an A in the course? It's very simple. You do the reading before class, you come to class, and you do the homework. And I guarantee if you do those 3 things throughout the course that you'll do well in the course. And I've said this almost every time I've given the course, and nobody has ever told me that I'm wrong. And so do these 3 things. If you don't get an A, then you can come back and talk to me about it later. The assignment for the next class is to do problem 2 of chapter 1, which I've repeated right here. So, and that's to think beyond what I've talked about in terms of what is biomedical engineering. To think a little bit more about biomedical engineering projects that you've encountered in your life or that you have some experience with, and then to think beyond what information I've given you in the chapter or in this lecture to say what products of biomedical engineering do you expect to become routine in the next 50 years. So spend 10 or 15 minutes thinking about this and write it down and bring your responses to class in the next period, and we'll talk about that. And so at the end here of this first lecture, where I've gone some way in trying to tell you what biomedical engineering is about, I thought I would try to relate it in a different sort of way. And you've heard this poem, right? London Bridge is falling down. Everybody's heard this poem. You played the game. I don't know if there's video games now if people play games like this where London Bridge is falling down. But this is a picture of London Bridge. It's an interesting bridge which is important in the history of London. Bridges have really changed our society and allowed us to get from one place to another in ways that we couldn't have gotten to easily before. One of the interesting things about London Bridge is that it's now no longer in London. It's in Arizona. You can see a palm tree here. But when they reconstructed London Bridge, they moved the old London Bridge to Arizona. Some guy bought it. That must be an interesting story, but I just have it here. And I think the poem tells you something about engineering, right? If you go through and the problems of engineering. And in bridge building, we're well advanced in understanding what are the problems with building bridges and how do we overcome them. For example, one thing that could happen is that you build it up with wood and clay. You pick the wrong material for a bridge, and it will not stand up to the forces of nature, right? It will wash away. And so you've got to pick the right materials in order to build a bridge. So you pick a better material like iron and steel. That makes a better bridge. We know that now because we have experience with bridges. But still, your bridge might fail. It might fail for a different reason. It might bend and bow. That is, it's not the forces of nature like the movement of the river that's knocking the bridge down, but it's just the failure of these materials over time, right? That they don't last as long as they might. And so you build it with a material like silver and gold, and then you encounter the problems of society, right? That your bridge might get stolen because somebody thinks they have a better use for silver and gold than your bridge. And I would say that in biomedical engineering, largely, we're still at the stage where we're trying to understand how things work and how they fail and what materials are the right ones. We're maybe where civil engineering and bridge building was 100 years ago. And that makes it, for me, a very exciting time to study this because the problems aren't solved in the way that bridge building is largely a solved problem now. The problems like the artificial heart are still unsolved. There's still room for innovation, still room to learn from what hasn't worked before, to learn from science, and to design something better. And so one of my purposes of this course is to get you, whether you study biomedical engineering after this or not, excited about the subject so that you start thinking about how you could innovate in this area where lots of problems are still left to solve. So I'll see you on Thursday, hopefully.